invitation to present this program and the data that I will talk about. So within the next 30 minutes or so, I'd like to introduce you to a field that I believe most of you will not have a direct contact to, and that is the field of oncolytic viruses. And the name already implies it. It's been used to treat cancer. And to start out with, what you see is the company called Grifols behind it. Grifols is actually a global healthcare company, mostly known for its plasma products, so it's not a typical pharma company. But Grifols decided 5 years ago to create a subdivision dedicated exclusively to the ATMPs, meaning to gene and cell therapies, and the subdivision called Gristle. What we do is we don't engage directly in investigation, but what we do is we act like an investment fund. We invest in companies and startup companies that have technologies in this field. And so what I'm going to present to you today is the work of one of these investments that we've realized, and the company is called VCM Bioscience. It's a company that's been derived, that was spun out of the Catalan Institute of Oncology, or ICO, and one of the founders spent most of his career in the virotherapy field, meaning using viruses to treat, to use for cancer treatment. His name is Roman Alemani. And so in 2012 we invested in this company, and VCM Bioscience uses this platform of adenoviral oncolytic viruses to develop products targeted for different cancer indications. The technology that I'm going to present, called VCM-01, has the orphan drug designation from EMA for pancreatic cancer. Obviously it has a certain patent protection behind it, and as mentioned before, that's one of the aspects for industry that is of importance. And currently is in 2 phase 1 clinical trials, and I'm going to present you some data from those trials. There is a follow-up product called VCM-02 in the preclinic, but I'll focus simply on the first one. So what is oncolytic virus? What is an oncolytic virus? The use of a virus to actually combat cancer is nothing new. It's been described already in the 50s, where it was discovered that patients with a cancer had a regression due to a viral infection. So viruses replicating in tumor cells have been discovered in the 50s, but obviously it wasn't until molecular biology allowed us to develop tools to be specific and target and manipulate these viruses and to understand virus biology on a molecular level that these tools actually could advance. Now, the virotherapy, as the definition is there, is using replicant-competent viruses that are restricted in their replication to tumor cells. Right? So the important part here is the virus is replicating. Now, depicted here, do we have a laser? Okay. Depicted here, basically, if you start out with an oncolytic virus, the virus usually can infect various types of cells. The specificity of the virus defines which type of cells it can infect, and as we all know, various viruses infect various types of organs. Now, the difference is that the oncolytic virus usually is manipulated in a manner that replication only occurs in the tumor cell. So, once the replication then occurs, you obviously have a lysis of the cells and you have to spread it. Oops, that was that. So, the concept is that you have a selective killing of tumor cells. The replication does not occur in healthy cells. You generate, each time the virus replicates, thousands of copies of this virus, so it's a self-amplifying product. And on top of it, the cells that are infected obviously express viral genes, so they elicit immune response. And there is also the belief and theory behind it that the lysis of the tumor cells will release tumor antigens that so far have not been recognized by the immune system, and that can be presented then via the antigen-presenting cells, so macrophages infiltrating the tumor, and can stimulate an immune response towards with cancers. Right? So we look at an oncolytic virus. Obviously, one of the aspects that is of importance is how much that this virus actually penetrate the tumor. The tumor in itself obviously is more like an organ. It's a complex structure. It's not just an accumulation of the same type of cells. We all know there's vascularization. We have a lot of fibroblasts in it. There's a certain type of mesenchymal stem cells that are associated. The cancer stem cells may be there. And there's a lot of matrix proteins keeping the whole structure together. So we also know that the tumor is not a tumor, meaning the pancreatic tumor is different from a breast cancer tumor, from a colon tumor. So each tumor has certain characteristics that are also manifest itself in the architecture of the tumor. Right? And so if we look at it at an epithelial tumor that we have here, one of the characteristics is that you have this invasion of the tumor cells, you have the blood vessel, and one of the problems that a lot of chemotherapeutic agents face is actually reaching the target, meaning the stroma of the tumor and the interstitial tuber of the tumor often prevents the penetration of chemotherapeutic agents or of antibodies for that matter as well to actually be efficacious. So the architecture of the tumor causes very often a problem in treating adequately the tumor with the products that are available. So the same holds true, obviously, that the architecture limits to a great extent the infiltration of T cells and of macrophages to elicit an appropriate immune response. So now, what is the VCN1 technology? What is the product? Now, the product is an adenovirus. Probably 80% of us here, we have antibodies against adenovirus. Adenovirus is a common virus, especially the adenovirus serotype 5. It produces a slight cold, a little bit fever, infects usually lung tissue. So during our lifespan, most of us actually have an infection. The advantage of the adenovirus is, especially the adenovirus 5, it's a virus that's been around in the science for decades. I think every single base pair is known, it's been sequenced. There's a lot of knowledge of knowing which viral protein does what, what's the purpose, what's the effect. The adenovirus has been used throughout the last decades in the clinic for different trials and different advances. There's a lot of knowledge about the response, the human response against the viruses, about its toxicity. So a priori, we know a lot about this virus. Now, the candidate has incorporated several modifications compared to the wild type. For once, what I was mentioning before is the tumor selectivity. Now, the adenovirus infects a variety of cells. The infection occurs via the so-called CAR receptor, as well as integrins, both of which are expressed abundantly on tumor cells. But there's a deletion in the promoter plus deletion in the E1A protein of the virus, which interferes with the retinoblastoma pathway. So basically, what happens is that these cells can only replicate, this virus can only replicate in cells that have an effective retinoblastoma pathway. One of the mechanisms that every cell has upon a viral infection is upregulating RB, because RB interacts with E1A and then downregulates the whole replication part. So this is blocked. So meaning that replication occurs in cells where RB is defective. Then there's some modification against the fiber. So here's a small virus depicted of this characteristic structure, and you have these knobs, which are called the fibers, and they are important for actually the interaction with the cell surface integrins. So there's been a slight modification in this fiber knobs that have been published and have been shown to reduce the interaction with hepatocytes or with the liver, because one of the major uptakes of adenovirus that we administered intravenously is a clearance in the liver. And on top of it, there's been an enzyme-induced which is hyaluronidase, and is one of the hyaluron is one of the stroma matrix proteins secreted and very abundant in pancreatic cancer. So, which again, the idea behind this is destroying the tumor matrix to facilitate the spreading. So again, we have these three different sections: the selectivity and replication of the tumor cells. There's a certain selectivity with the RGB, trying to avoid the liver targeting and retention, and thereby increasing the disponibility towards the cancer cells. And then within the tumor, the actual expression of the hyaluronidase enzyme to degrade the matrix. And the way that the virus has been constructed is only upon viral replication is this enzyme being expressed. So we do need to have the viral replication in order to have the enzyme expressed in order to degrade the matrix. Right? So the mechanism of action behind it is twofold. It's the tumor killing of the replication, the lysis of the cells via the viral replication, and degradation of the stromal matrix, which will then allow A, a better spreading of the virus, and B, the entry of other pharmaceutical agents. So in the case of pancreatic cancer, it's known that most of the pancreatic cancers, the tumor cells are not chemo-resistant, but it is actually a mere problem of the chemotherapeutic agents to enter the tumor. The tumor has a high interstitial tumor, and I'm tempted to say all, but at least the data that I know is of the tumors that have been taken out, all of the tumor cells were reactive towards chemotherapeutic agents. So, which clearly indicates that the chemotherapy that's been administered is not very effective. And it's not effective because the cells don't respond, but simply because it doesn't get there at the concentration that it needs to get. So, now, with all this concept, where are we at? So, what does it do? And obviously, as I was indicating, there is a lot of preclinical work, so the lab work, to actually show that what your hypothesis is holds true on an experimental basis. Now, on once, I don't know if you can see it very well, but there are two viruses, predecessors of the VCN1, which are very similar and were used in these experiments. And the difference between the 15 and the 17 basically is the 15 doesn't express the hyaluronidase, so the stromal matrix degrading enzyme, and the 17 does. Now we have here, these are images of tumors from mice that were obviously then taken out. This is a melanoma tumor cell line, and this is a pancreatic tumor cell line. Again, here's the 15 without the hyaluronidase, and here's the 17 with the hyaluronidase. And we're looking at viral replication, staining for the virus that is replicating. And I think, I hope that you can see it quite clearly, that the actual distribution within the tumor increases drastically upon the degradation of the tumor matrix. So one of the concepts of a better degradation, a better distribution of the virus, at least in the animal model, holds true. In a different experiment, different analysis, the VCN1 virus, so the product, was injected. It's a pancreatic tumor cell line at B9. And again, at day 79, the tumors were taken out and were basically stained against hyaluron. So the matrix protein that is in this tumor present, and what we can see is in the untreated tumor we have a strong staining of hyaluron, of the matrix protein, whereas in the tumors that were infected with the virus, and aside from the fact that you clearly see the size difference, what is reminiscent of the tumor expressed very low amounts of this matrix protein, and this is just a positive control. Now, last not least, there's obviously the aspect of efficacy and the synergism that I tried to sell to you, meaning once the stroma is degraded, how does it work with any other chemotherapeutic agent? Now, again, probably hard to see. It's a model, NP9 model. It's again pancreatic cancer model. Now, this is the line of the tumor that is untreated, so it's just PBS administration. Then you have gemcitabine, which is the typical chemotherapeutic agent that's been used prior to Avaxana. Here is our virus, injected alone. So already on tumor size more efficacious than the chemotherapeutic agent. And then combining the chemotherapy and the virus, what we clearly see is a synergistic effect, meaning that those two actually work together much more efficaciously than each treatment alone. Now, based on this, and obviously there's a lot more data, and there's also a lot more regulatory preclinical work that had to be presented in order to get these trials approved, VCN started 2 phase 1 clinical trials. And basically the 2 phase 1 clinical trials are with the same product, VCN-01, so the adenovirus in pancreatic cancer. Just one is an intravenous administration and one is intratumoral administration. So the intravenous administration is a single administration, and the typical design for phase 1 is dose escalation. To define basically the maximum tolerated dose that you can administer, but also in the end to evaluate obviously the toxicity and biomarkers, and we'll come back to that. Additionally, as I mentioned before, most of us have neutralizing antibodies against adenoviruses, so the administration of an adenovirus intravenously is not so much an aspect. It's not so much an aspect of toxicity if we have a high titer, but it's an aspect of efficacy. So if you have a high titer and you eliminate basically the product that you want to administer, how can you monitor or try to get efficacy if you basically eliminate the product by itself? So this plays a role in the intravenous administration, and thus we monitored prior to the administration the neutralizing antibodies, and we decided we have a cutoff of 1 to 320. It's a neutralizing assay that we set up. So calculating basically on the dosage that we administered, we defined a cutoff level, which allowed us so far to basically have 60% of the patients that we included to be treated. Now the intratumoral administration is repeated. It's 3 administrations, and again it's a dose escalation. And this dose escalation then will be in combination with gemcitabine and Avaxana. Originally it was for locally advanced pancreatic cancer, but that now changed. And again, evaluation of tox, biomarkers, viral shedding, etc. But again, I think there were 2 main concepts behind it. One is the intravenous administration clearly gives us the limits on the fact of, or on the basis of toxicity. What is the maximum dosage that we can administer? So the maximum tolerated dose level has been defined. In the intratumoral administration, we're not so much concerned about toxicity. It's also been measured, obviously. But what we're monitoring, or wanted to monitor here, is more the biological effect, and see if everything that we see in the animal we can actually observe in the tumor. Because if you inject in the tumor, at least you know that the virus reached the tumor, so you're sure of that. So what is coming next is trying to understand if the replication works as you've seen in the animal models, what biomarkers can you extract. Now, the data that I can show you is we finished one part of the intravenous administration trial, and this is basically the dose escalation phase. The dose escalation phase was done mostly in patients with colorectal cancer that had hepatic metastases, and so to speak were failing any type of treatment that so far had been used. And in that case, what is very typical of a phase 1 clinical trial in cancer is you get the worst patients you can imagine. And sorry to say, for those patients, obviously their life expectancy by definition is rather low and bad. And of all of the patients that we've treated so far, I think there are only 6 still alive. So again, the exclusion criteria based on the antibodies, and then we had different dose escalations. So 10 to the 11th viral particle up to 10 to the 13th. And the highest dose that we used 1 to the 13th is also the maximum tolerated dose level, so we could administer the highest dose that we hope to administer. So this is basically the first part that we concluded. Now, the second part of this phase 1 study basically is conducted in pancreatic cancer patients only, because it is not a monotherapy anymore, meaning only the virus, but is now being combined with the Avaxan gemcitabine treatment, which is the standard of care today in pancreatic cancer, and which allows us to recruit basically cancer patients in first line. So the advantage is that the second phase that is basically started now allows us to have a pure optimal cancer population. These are patients that just recently have been diagnosed with pancreatic cancer and would receive now the gold standard, the standard of care, which is Avaxan and gemcitabine. And if they agree to participate in the trial, they would receive on top of the normal chemotherapy the viral administration. So again, it's a dose escalation. It follows the same concept. So we go down one dose level from the maximum tolerated dose, add the chemotherapy agents. If there are no dose-limiting toxicities, then we advance after 3 patients to the next level, to the maximum level that we define here. Have 6 patients, and the protocol allows us to expand to 9 more patients, so that we actually could have 15 patients treated at the highest dose, which at least is a nice number to monitor biological activities in the biopsies that we hope to get from these patients. And I don't want to talk about efficacy, but at least to get an idea of if we administer the virus and the chemotherapeutic agents, do we see any effect, any beneficial effect on that. But again, the important part for us is that the design of the protocol allows us to basically recruit pancreatic cancer patients only in the first-line treatment, simply because the chemotherapy is only approved for pancreatic cancer, so there are no other cancer patients that would receive Avaxan and gemcitabine at the current stage. Now, this is a summary of the adverse events that we noticed in the first phase of the intravenous administration. And basically, the important key messages, there's a variety of adverse events that have been noted, but they're all in line with what we've seen in the preclinical data. So there were no ugly surprises in the clinic. We've seen in the preclinical models all of the symptoms that were shown later on in the clinic. It is also in line with what has been reported with other adenoviruses. So our adenovirus behaves on a toxicity level very much so as other adenoviruses that have been injected in human. The most common side effect is mild fever. It can go up to 39, I think there was one patient up to 40, but it's transient. And this is the important part. All of the associated toxicities are transient. They're produced within the first couple of days. It's fever. There are transaminases that go up because it's been basically the effects on the liver. And after a few days, all of these symptoms reside, and there are no long-term effects so far observed. All parameters go back to normal. And we only had one dose-limiting toxicity reported, and that was a patient that was mentioning that you get kind of like the worst patients you can imagine, and he had a metastasis in a cerebral metastasis, and it caused side effects that obviously at the co-administration of the virus then is always difficult to distinguish in how far your virus has been participating in that or not. So automatically it was then concluded DLT because the transaminases were very high, and he had other issues. So the intratumoral administration, basically again a dose escalation study. This was the original design. In the intratumoral administration, we did not go for the monotherapy, but it was always combined with chemotherapy. And as indicated here, it was originally designed to only be co-administered with gemcitabine. And due to the entry of Avaxan in the in the treatment and in the hospitals. Basically, we could not recruit any patients simply because the MDs or the centers would not recruit patients because they were receiving the first-line treatment, which is the Awaxana gemcitabine. And so we changed the protocol to include in the chemotherapy the Awaxana gemcitabine treatment. Also, since we concluded the toxicity in the intravenous administration, which is the worst route of administration, so to speak, we shortened this dose escalation. We modified the protocol to only go for the last 2 dosages, the 10 to the 11th and 1 to the 12th, and adding the chemotherapy agents. The protocol has been accepted by the Spanish authorities, and again, we start now with this part as well. Again, it's a first-line treatment in pancreatic cancer, with the difference that obviously they need to be diagnosed to have accessible, ultrasound-guided accessible tumor, which is not the case in our cancer patients, in our pancreatic cancer patients. So the recruitment will definitely be a little bit lower, but it will give us a good way to actually obtain biopsies and to verify in how far the virus actually is being replicating, and also over how much time it is replicating. If the immune system is basically eliminating everything, or if the virus can be found for several weeks. So within a year, hopefully, we will have some more data. Now, where do we go from here? And I think within the last couple of years, the oncolytic virus field was a niche sector, or niche product that no one really cared about, and pretty much all of that changed due to the huge success that the inhibitors had, the immune checkpoint inhibitors had, especially the PD-1 checkpoint inhibitor in melanoma patients. And if any cancer congress, any even gene and cell therapy congress where there's a combination of immunotherapies, there was only one hot topic, that was the anti-PD-1, the CTLA-4. So the immune checkpoint inhibitors is the hot thing now in cancer treatment. Nevertheless, the field recognizes that it clearly cures a certain percentage of patients, and I think the current combination of CTLA-4 and anti-PD-1 in melanoma patients, metastatic melanoma, goes up to 50%. Nevertheless, there is still a patient population that does not seem to respond, and Big Pharma is looking for additional reagents and products that act as a supportive agent. And so Big Pharma actually turned to the oncolytic virus field, and there's quite a strong interest right now in data that's been produced with oncolytic viruses. There's even in the US a product that's about to be approved by FDA. It's presented by Amgen, who bought the company called BioX a couple of years ago. It's based on the herpes virus. It's injected intratumorally into a melanoma, so it's not intravenous administration. So what they are interested in is a virus that could be administered intravenously. And again, a little bit what I was mentioning before, the idea behind it is that obviously if you have a virus that then enters the tumor, replicates in the tumor, opens up the tumor, in combination with the checkpoint inhibitors, would allow for a synergistic effect that may be able to target now the patient population that do not respond to the checkpoint inhibitor alone. And so the increase of infiltrating tumor lymphocytes in combination then with the checkpoint inhibitors and the oncolytic virus is what everyone is hoping for now to produce results within the next couple of years. That may even give a higher push and result as an efficacious product for a lot more cancer than just melanoma, but a lot of the other incurable cancers.
May I ask for a couple of questions? Thank you. Unless you want. You had a question?
No,
okay. I was wondering, from what I understood, you have a very good tolerance with IV administration. Currently, you are investigating intracerebral, but not all patients have targetable pancreas cancer. So you cannot treat all the patients because you cannot really identify.
The two aspects. In the intratumoral administration, obviously the tumor needs to be located and the diagnosis needs to identify the tumor as injectable, right? As administration by ultrasound endoscopy. So this is a small percentage.
Small percentage.
Well, small. Probably right now about 40%. What we hope is maybe administrable. But again, this is purely a diagnosis based on the physician that clearly needs to indicate, okay, this patient could be reached via ultrasound endoscopy or not.
Okay.
The intravenous administration, the limiting factor basically are the neutralizing antibodies.
Okay.
So, and again, the effect of what is the limit is hard to say. We don't have any reference data. What we wanted to do is we wanted to avoid that in the end we have patients with really high titers where we believe that a priori little or none of the virus actually had a chance to reach the tumor, because how to interpret this data, also based on toxicity, is this, would distort the whole interpretation. So we try to be as clean as possible. And calculated the limit that for us was acceptable.
But did you measure neutralizing antibodies?
Yeah. So prior to administration of the product, each patient, it's a blood sample, it's an assay that's done within a week. So prior to the administration of the virus, they undergoes the screening, and of the screening, 60% of the patients so far were below the threshold, so 40% were excluded.
Yeah.
I have a more scientific question. So in preclinical studies, I'm not sure you demonstrated that this is the oncolytic effect of the virus that is killing tumor cell proliferation, or rather the effect of hyaluronidase activity.
No, it's the virus.
It's the virus.
So again, you can do cancer cell lines. There's a whole battery of cancer cell lines in vitro where you just simply infect with the virus, and then the virus replicates in cells.
Yes.
And if you only administer the hyaluronidase, it doesn't destroy anything. It doesn't destroy the cancer. So if you were to inject into the tumor just the enzyme, and it's been done for patent reasons, you do not destroy the tumor. So the hyaluronidase only degrades the matrix. It doesn't degrade the tumor. It's also been demonstrated in contrast, for example, to collagenase, that the hyaluronidase is not sufficient to actually liberate the cells and thereby increase metastasis, for example. So all that you do is it's a little bit like a Swiss cheese. So you create holes in the whole tumor network, so to speak, which facilitates the spreading of the virus. And what we've seen is it facilitates also the entry of the chemotherapy.
So this is my question, because the combination might be useful if you just destroy the matrix with hyaluronidase and you add chemotherapy. So I don't know if you did the experiment to have the virus, but just with hyaluronidase activity and not retinoblastoma.
The problem is, how do you administer into the virus the hyaluronidase? How do you grade the matrix in a homogeneous manner? So the virus, the replication of the virus basically allows for much better destruction of the matrix within the whole tumor mass than an injection. You would have to inject the enzyme, right? There's a company that actually uses the recombinant protein and administers the protein intravenously, and it has toxicity effects. So there are side effects. So having anything that you can administer locally, restrict locally, on the toxicity level is always advantageous. It always gives you less side effects than things that you have to administer intravenously, right? Systemically. And simply the distribution coupling the hyaluronidase with the viral replication, again, allows us to have the effect of the degradation of the stroma restricted to the tumor, because replication only occurs in the tumor, right? And on top of it, it's an additive effect, in the sense that you lyse the cells, you already create certain certain holes, if you want, and then in this environment, on top of it, you have the enzyme that allows the degradation of the surrounding tissue, facilitates the spreading of the viruses that you release when the virus lyses it. So now, at which moment, which of these aspects has which importance and which weight is nearly impossible and difficult to distinguish.
Can I continue in the discussion with retinoblastoma targeting of cancer cells? So are you sure that all pancreatic tumor cells have the retinoblastoma pathway overactivated? Probably you have different grades and different type of mutations. So in your patients, did you check whether they have some mutations or not of the retinoblastoma pathway?
We didn't check.
I think
so
you can, because
in the biopsies, obviously, when we take out, we can look for, we can check that, and it's a good point to correlate the replication. So what's been described, what's been known also from primary tumors is that.
But it could be nice.
No, there could be
some responding patients with this mutation and non-responding patients without this mutation could be very nice and indeed.
So the issue is basically it's a dual aspect. It's P53 and RB. So it's not a solid as only RB, but it's a combination of P53 RB that plays a role in the replication. So to have a tumor cell line that is non-responsive to the replication. We don't expect to find it, but in the biopsies, it's clearly something genetic screening to observe and to monitor and to evaluate if there are tumors that then did not respond because of application. But again, the hypothesis right now is that this is not a limiting factor. So far, what's been described for known pancreatic cancer is that this pathway is altered and would allow.